In this video, we will learn about designing primers and generating fragments for the Gibson assembly. To recap, Gibson assembly is a seamless molecular cloning method for joining of multiple DNA fragments. This happens in a single tube as an isothermal reaction and simplifies the process of molecular cloning. This technique was developed to create exceptionally long DNA constructs in vitro. From then on, it has proven to be a robust technique for combining multiple long and short fragments. Here you will see the general workflow of the Gibson assembly. This method is based on assembling fragments containing overlapping ends. The vector and DNA inserts are first amplified to introduce the overhangs. Then, they are combined with a master mix containing 3 enzymes. The reaction is incubated at 50 degrees Celsius, usually from 15 to 60 minutes. The result is a fully sealed plasmid, ready for transformation in the competent cells. To perform Gibson assembly, we will need to prepare the inserts and the vector. The first step is to design the primers. It is important to understand that Gibson assembly requires fragments that contain overlapping overhangs, and we will learn how to create them. The first thing we will focus on is the preparation of insert. The inserts are usually prepared by PCR amplification. Here, we have an example of an insert that is a gene encoding esterase we want to characterize. This gene is present in the genomic DNA of a bacterial strain. Using specifically designed primers, the gene is amplified during a PCR reaction. This results in the addition of homologous ends. The ends are needed for the joining of adjunct fragments, which is, in our example, the vector. For PCR, we use the high-fidelity polymerase. With this, we ensure that the error rates during PCR amplifications are low. The addition of homologous nucleotides is ensured by carefully designing primers for PCR amplification. The primers contain 2 parts. In this example, we have the template-specific part and the vector-specific overhang. For the template-specific part, standard PCR primer design guidelines should be followed. Primers should anneal well with the targeted DNA and meet the standard requirements for optimal PCR amplification. For the vector-specific part, nucleotides are overlapping vector sequences exactly at the position where we want to introduce the insert. The length of homologous overhangs depends on the insert length and the number of inserts in the assembly. If the fragments increase in length, it is necessary to increase the length of the overlapping ends. With the increase of the number of fragments in the assembly, the length of overlapping tails should also increase. For a simple Gibson assembly reaction, having a vector and 1 insert, overlapping ends from 20 to 40 nucleotides are usually sufficient. The next step is to prepare the vector. In general, there are 2 ways we can prepare the vector. One way would be to linearize the vector using restriction enzymes. However, we will not cover this approach during this video. The second, more common approach is to generate linearized vectors by PCR amplification. In that way, backbone of the vector is amplified using specific primers. Usually, these primers contain 2 parts, the vector-specific part and the insert-specific overhang. After the PCR amplification, the vector fragment contains insert-specific ends. Next step is to incubate PCR products with DPO1, the restriction enzyme. This enzyme specifically cleaves methylated DNAs. In this case, the enzyme digests residual plasmid template. With this, we ensure that we do not have vector template while transforming into the competent cells. If this step is skipped, we can have quite some colonies carrying an empty plasmid. Finally, we have both amplicons containing overlapping ends ready. The PCR results are visualized by running an agarose gel electrophoresis. Amplicons can be cleaned up with the PCR cleanup kits. If multiple bands are visible on the gel, it is recommended to perform gel extraction. The next step is to measure the concentration and quality of the generated fragments. For this, we use nanodrop spectrophotometer. It is an instrument used to measure the concentration and purity of nucleic acid, such as DNA or RNA, and proteins. It is easy to use, requires only small sample volumes of 1 to 2 microliters, and provides us with quick results. The device measures absorbance at specific wavelengths. Nucleic acids absorb at 260 nanometers due to aromatic bases present in DNA and RNA. Proteins are measured at 280 nanometers. Proteins absorb due to aromatic amino acids such as tryptophan or tyrosine. Contamination, such as organic compounds, for example phenol or carbohydrates, absorb at around 230 nanometers. Based on the absorbance at specific wavelengths, the nanodrop calculates the concentration of our sample using Beer-Lambert law. For cloning purposes, we aim to have high purity samples of nucleic acids. Pure sample characteristics include a clear absorbance peak at 260 nanometers, indicating the presence of nucleic acids. We aim for minimal protein contamination. This is visible as an absence of significant absorbance at 280 nanometers. Also, we aim for low organic compound contamination with minimal absorbance at 230 nanometers. On the slide, you can see the key absorbance ratios we follow and some guidelines of ratios indicating highly pure DNA.